Assalamualaikum warahmatullahi wabarakatuh and a very good day to all the examiners and chairpersons. Today I would like to present my research regarding the oil palm residue biodelignification by Bacillus cereus ligninase from Coptothermus curvignatus gut for handshake production. My name is Sharfina Mutia Sharifa and I am a master candidate from the Faculty of Engineering Technology UTHM. My research work was guided by my supervisor, Prof. Madhya Dr. Angzasari binti Muhammad Kasim, and also my co-supervisor, Dr. Muhammad Firdaus bin Abdul Wahab from UTM. Let's begin. For my presentation content, there will be 6 parts. Each part represents a chapter from my thesis. First off is the introduction. Secondly is the literature review. Third is the research methodology. Fourth is the results and discussion. Fifth is the conclusion and recommendations. Sixth is the closing. As you all know, you are now looking at a video where there are students doing group assignments in the library. This indicates the usage of paper in academic region, large offices, and also in everyday life globally. But wait, you might be thinking that in this era everything goes online. Logically, all paper products should have decreased since the global internet breakout. However, staying at home or out of office, paper are being used in different ways, such as the essential use of paper products, e-commerce business, paper products replacement for plastics, which significantly rise today's paper production. The largest industry of the world, the fourth largest industry, faces its challenges. The conventional methods using toxic effluents, wastewaters, land clearing that causes deforestation and irresponsible industries contributes towards the global warming or even climate change. Furthermore, the use of huge energy consumption during pulping due to the recalcitrance of lignin also is the problem. Furthermore, the government stricting the rules of lands and forests also has affected the supply of foods, and also due to the increase of human race, which require more demand in all needs. Researchers all over the world are required to make a change to save the environment by cutting the production costs, reducing the energy use, and friendly environment industry. In 1978, Kirk has introduced as a new technique of green pulping through biodelignification using the world's microorganisms. This is up to date has been the continuous work of research to find the best act of microorganism. Dwelling with the alternative resource or pump biomass being used as an alternative for the expected future use. The world paper and paperboard has increased steadily over the past years. Especially for Asia, it has become the highest or top producer and consumer. Asia has a production of about 47% and the consumption about 48.4%, while it is followed by the SAPI, North America, Latin America, other European countries, and the rest of the world. The global total in 2017 production and consumption reached more than 400 million tons, which the consumption itself have a higher number, which is 423.3 million tons, compared with the production, which is 419.7 million tons. For the annual addition, both production and consumption has increased for about 1.7%, and the growth per annum itself since 2009 has increased about 2.1% and is going to be predicted for an increase about 6 million tons per year. Thus, the problems are addressed in this research is regarding the resource problems, where demand has been increased due to the e-commerce and variety of paper product replacing the plastic products. The shortage of wood resources where deforestation being caused, and also for more oil palm plantation is being used in the uh, area, woody plantation area. And for the import is because non-feasibility, and also the local source actually has less quality and have perhaps less amount of land for the wood plantation. Thus, an alternative approach introduces oil palm biomass as a replacement where it is high in fiber for paper production, economically friendly, and also contribute less waste and pollution. For another address problem is that the industrial process problems indicates that chemical actually produce pollutants, and it costs more for the waste management. And for the mechanical problem, it over soften the fibers, and it's costly due to the high energy consumption. For biological fungi part, it has a long fermentation period, sensitive and difficult to regulate. Thus, an alternative approach for this is to use a bacteria for biopulping, where it is highly efficient in degrading lignin, economically industrial friendly, and contribute less pollution, promote greener technology. For the aim of this research is actually to elucidate the lignin degradation by the ligninase expressed by the bacteria of Bacillus cereus from the gut of a termite, wherein it actually will provide an alternative for the application in the pulping, industrial application for the pulping, and it also used for the production of handsheet using the oil palm residues. So the first objectives is actually to express, isolate, and identify the lignin degrading enzyme from the bacteria of Coptothermus perfignatus gut bacteria, which is the Bacillus cereus. And secondly, it is to analyze the biodelignification ability of lignin degrading enzymes on treating oil palm residues based on the chemical composition. And lastly, for the objective is to evaluate the handsheet produced from the biodelignification activity treatment on oil palm residues, testing on its physical, mechanical, and also pulp properties. For Chapter 2, Non-Wood Oil Palm Biomass, it is an alternative resource for the wood replacement due to wood resource problems and a 5 years of cycle compared with the non-wood, which is only 3.5 years of cycle. This also contributes to the incline incomes for the farmers. And for that, non-wood actually is being used for the production, but it is less than 10%. And the average annual production in 2018 is 19.5 million tons, which the waste itself caused a major disposal problem. And for that, the oil palm leaf itself has a good cellulose content which is acceptable for the range of paper and pulp industry. And with that, low lignin content. For the oil palm trunk itself, the OPT have 80% OPT sap that are highly in cellulose. And for that, the trunk also has a potential for paper and pulp and has been discovered for delignification study using the Aspergillus species. And for the EFP itself, it is successfully pulped by an alkaline method, and it's known for its good printing method. And for the biopulping and fermentation, biopulping itself utilizes natural degraders for the wood chips and also lignocellulose materials, which is prior to the mechanical or chemical pulping. And for that, biopulping is actually compatible with other pulping techniques. Here is a table where it's shown the differences between mechanical, chemical, and pulping, the comparison, where biological pulping have more advantage compared with the mechanical and pulping methods, which possibly can produce higher paper quality and fiber strength. And for the submerged fermentation technique is actually used for the extracellular enzyme, and this is also actually simpler for the engineers in industrial scale for its management and sterilization. Thus, for the fermentation process, Bacillus is more skewed or fond with the SMF fermentation method compared with the SSF, which is the solid state fermentation. And here shown in the literature review that bacteria for Bacillus species tend to produce LIP and lactase, which is the lignin peroxidase and lactase. And for those, the insect animals, Coptotermus or termites are known for ligninase enzymes, delignification enzymes. So this is a potential use for paper and pulp industry. And for the lignocellulose, I've been talking about lignin for a lot, and now I would like to explain that lignin is actually a structure where it binds all the fibers together and perform its plant rigidity. And for that, the lignin structure itself is very complicated. It's an complex aromatic compounds, and for the hemicellulose and cellulose structure is uh, having a less, or I mean not so complex a structure compared with the lignin. And for the scope in this research, there are three types of three different scope for the objectives 1, 2, and 3. However, in objective 1, empty fruit punch is the only oil palm biomass used, whereas for objective 2 and 3, all three types of biomass is being used. Moving on to chapter 3, the research methodology isolates three objectives into three parts of the experiment. The first is the expression isolation identification of LDE, where enzyme extraction, partial purification, quantification, analysis of the protein sequencing are being experimented. And for the biodelignification ability analysis, the bacteria culture, SMS treatment analysis for its content analysis of the lignocellulose, all four contents are being analyzed, and for the handsheet production is for the biopulping, mechanical pulping, and the handsheet production. So for the objective 1, the bacterial culture and enzyme expression are being laid out for this experiment, where the Bacillus cereus are incubated in the universal bottle to increase its amount, and using the Macfarlane to actually identify the amount of protein or enzyme expressed. And so far, the SMS treatment are using 37 degrees Celsius, 6.5 pH, and speed of 120 RPM in 7 days of fermentation. And afterwards, it is extracted in the centrifugation and also partially purified using precipitate against the ammonium sulfate, dialysis tubing, ultrafiltration. And the end is that it is analyzed for its protein quantification using Bradford assay. And for that, the identification is using the SDS page SDS page electrophoresis for the molecular weight determination, and also for the protein sequencing sent to the Proteomics International. And for the second objective, The research methodology are using TAPI standard and also the chloride and Kushner-Hofner method. And also it is continued for the biopulping process for the objective 3, which is also continued with the mechanical pulping, where there are 4 steps for the mechanical pulping just to disseminate or actually break down the chips, the non-wood chips. And also for the handsheet production, it undergoes 8 steps. First off is the pulp weight and moisture. It goes to disintegration, correction, and also handsheet making up until the pressing process and stacking of rings to produce the handsheet. And continued with the paper production in which was conducted at FRIM. The pulp properties was analyzed for the freeness and drainage rate, for the operational ability in industrial section, and also for the optical properties. It performed the brightness, opacity, and surface morphology analysis. And for the mechanical properties, there is thickness, grammage, tensile strength, tearing, and bursting strength analysis, all using the TAPI standard. So for the chapter 4, the first result for the BSA concentration graph for the Bradford assay standard was performed using the BSA, and also the absorbent was analyzed using the UV-Vis, and the absorbent and protein concentration were calculated using the R square and also the Y equation, and it indicates crude protein result to have a higher number of concentration compared with research lignin degrading fungi, which shows approximately only 0.69 milligram per mil. And for the protein sequencing result, the SDS page analysis was conducted on 3 different types of protein experimented, where the marker is put down in the first lane, and lane 2, 3 is for the crude enzyme, lane 4 and 5 is for the ultra filtered protein. And from there are 6 visible possible proteins that can be analyzed and was sent for sequencing, and it came out to have 11 different types of protein, where it gives a specified function, or the function itself contributes towards the synergistic mechanism of the lignin degradation. So the lignin itself as a sole carbon source corresponds with the stress response due to the decrease of carbon amount, pH, oxygen, phosphate, and also nitrogen. This actually activates the GAD system, the GABA, the shunt pathway that actually produces succinic acid. And the production of succinic acid, the succinic acid itself actually perform degradation of lignin. And for that, nitrite and nitrate reductase is also a part of the stress response, and the phosphate binding protein, the PSDS, and also the ABC transporter actually helps in the movement of aromatic compounds and also increases chances of degradation by exposing enzyme and help in ATP energy role for degradation. So another stress-induced oxidative damage also responds toward the heme peroxidase, where it is the prime lignin degrader enzymes, which is the LIP, MNP, and LAC, which is lignin peroxidase, manganese peroxidase, and lacase. And also the serine hydrolase identified was actually a part of lignin degraders, including the amidrohydrolase family protein that is susceptible for lignin degradation, as I and Y might be involved in this degradation, and the chemotaxis protein as a helping enzyme, also including the GMC family oxidoreductase and glycosyltransferase. The results for the objective to biodelignification ability analysis of the chemical composition laid out 4 different lignocellulose, which is the lignin, holocellulose, hemicellulose, and cellulose, were described in this slide. And for that, the untreated OPT substrates had the overall lowest reading for holocellulose, hemi, and cellulose content. However, the treated EFB itself has the lowest amount of lignin, which is more preferable, and it indicates a best pulp material for the handshake production. So the untreated EFB itself actually have the highest value of cellulose, and the percentage of lignin is actually higher than the treated EFB, which actually uh, reduce the bonding of fiber, where the untreated EFB were unable to perform as better as the treated EFB for the strength of paper. So the lignocellulose content comparison here is that the lignin content percentage decrease for all OPL, OPT, and EFB. So only EFB is actually considered for acceptable lignin degradation because it is perform a percentage difference of more than 10% compared with other substrate, which all the lignin content, actually the after treatment content, were actually acceptable for paper production. However, the percentage of degradation is not enough for a good paper or handshake. So for the hemicellulose content, the percentage difference here is that the EFB has had the most percentage decrease, and also followed by the OPL and OPT. However, the hemicellulose content decreased for OPL and EFB samples, whereas OPT actually inclined about 3%. So the high hemicellulose would probably increases paper quality and smoothness. And for the cellulose content, the percentage difference is here that the cellulose content increases for OPT but decreases for OPL and EFB. The decrease of cellulose content might be also due to the higher lignin degradation compared with other substrates. For the objective 3, the evaluation of handshake production, which the pulp characteristic shown that the freeness is actually an important aspect in machine operation, where 300 to 700 mil is actually acceptable for economical operation. So here for all substrate, it was shown that it has the range of between 300 and 700. However, the untreated OPL have the highest freeness level, but poor in its drainage time. However, for the treated EFB, the drainage time is the best. However, the freeness is not as good as the untreated OPL. However, it is still compatible and is acceptable for potential paper making, as high freeness with short drainage time contribute towards a lowest operational time. For the optical characteristic, the brightness and opacity percentage is described in this table. The untreated samples for the untreated EFB show the highest brightness, whereas for the opacity, the untreated EFB is shown to be the lowest opacity compared with the brightness. And for the treated samples, again, the treated EFB have the highest brightness with also the lowest opacity. The EFB treated samples actually had the best improvement compared with other samples, and this shows that it has high brightness and also high opacity, which gives an essential good paper product. For the untreated and versus the treated samples, the handsheet pictures shown in the slide that the OPL have quite a different appearance, where the untreated ones are unable to form a handsheet, while the OPT have two different color perspective due to the decrease of brightness, and also for the EFB quite similar color but have a higher opacity value. And for the surface methodology, OPL, OPT, and EFB are seen in one picture, where the OPL have the least distorted structure, which is not overlapping compared with the OPT and EFB, while the EFB surface itself have the most evenly covered structure for the 50 times magnification. And for the 200 times magnification, here it is shown that the EFB and OPT seem to have fiber bonding of the cellulose and hemicellulose structure with less gaps, and for the OPL structure had a very quite a big gap compared with other surface methodology characteristics. And for the mechanical properties, the tensile index for EFB treated was recorded 7.144 Newton meter per gram, which showed the increment of zero value, while the others are undetectable. Meanwhile, for the tearing index of EFB shown to be the highest as well, compared with other treatments, while the OPL recorded to be the lowest. And higher grammage and thickness in this study did not show support towards paper strength with poor tearing. However, tensile and burst value also have a low value, which did not give the support for higher grammage and thickness. Moving on to the mechanical tests, the comparison here shows that treated EFB have higher value compared with Acacia, Mangium, and Fine Shoot for its tensile. Compared with the tear and burst, it still have a low value compared with the others. However, it is still higher than the Fine Shoot and also higher than the Eucalyptus globulus for the burst index. EFB structure, or the substrate pulping process, was without refining and beating process. That's why it performed lesser lignin degradation. And also for the tearing and bursting index show to have a low value of tensile, which is important aspect in the strength and elasticity of the paper. It should have a higher value of tensile. In conclusion, for the first objective, the lignin structure breakdown is due to the synergistic mechanism of proteins identified in 11 types of enzymes. And for the second objective, the lignin reduction were capable for all three biomass substrate, and also the Bacillus cereus enzyme proven to degrade lignocellulose content, and the removal of lignin itself enhanced the reduction in the cellulose content. And for the third objective, pulp properties are crucial for the industrial performance, such as the value for freeness, drainage, brightness, and opacity for the EFB shown to be acceptable. And for the surface methodology analysis, EFB had the most bonded fiber with minimal gaps, and the mechanical strength for best result also belongs to EFB treated substrate. For the recommendation, future works on bacteria mechanism scope is important for the optimization of biopulping process. And in this research, experimental data were collected based on laboratory skill experiments so that future advanced submerged fermentation process needed to be conducted. And for the additional mechanical process, maybe perhaps optimizing industrial scale purposes, such as the refining and beating process. These are the list of the publications and the awards. And Alhamdulillah, thanks to Allah, beyond thank you, I would like to address my thanks for my supervisor, my co-supervisor, my parents, lab technicians, fellow lab mates, Forest Research Institute Malaysia, and lastly, the research grant from Science Fund. And that's all. To our future biopopping, thank you.